Hello, everyone. Thank you for joining today's webinar on risk assessment for nitrosamine contamination of peptide APIs. I'm your host, Lael Cheung. I'm a business development manager based in Boston. Today's webinar will be presented by my colleague, Matteo, Vice President of Chemistry, Manufacturing, and Controls at Bachem Americas. Bachem is a leading specialist in the development and manufacture of active drug substances. We focus on peptides and oligonucleotides. We strive to establish long-term partnerships with pharmaceutical and biotech companies by offering the best solutions for today and developing innovations for tomorrow. Our efforts contribute to the development of innovative medicines that are helping patients worldwide. Our business is built upon 5 strategic foundations, namely people and culture, innovation and technology, sustainability, customer centricity and service, and operational excellence and quality. We have some exciting updates to share on our efforts to build the world's most advanced manufacturing facility for peptides and oligonucleotides. Our first phase of expansion will be complete in 2024, and it will double our manufacturing capacity. When fully operational, this facility will employ over 300 people. We have earmarked over 350 million Swiss francs in investments, which will go towards applying innovative green chemistry techniques such as solvent recovery and wastewater treatment, along with Industry 4.0 practices to increase automation and innovative production technologies. This facility, when complete, will embody our commitment to sustainability and operational excellence. Before we begin, I'd like to go over some general housekeeping rules. This webinar is pre-recorded, however, we will have a live Q&A session at the end. So please enter your questions at any time using the Q&A function below. And with that, I'd like to hand over control to my colleague, Matteo.
Thank you, Lael, for the introduction. I would like to take you today through the agenda, first of all. So we are going to discuss initially what happened in between 2018 and 2021 in the pharmaceutical industry regarding nitrosamine. And then we will look into nitrosamine and what are the nitrosamine concerns and where they come from. Then we will look at the different way that Bachem has to prepare peptide, meaning we can produce peptide by solid phase peptide synthesis or by liquid phase peptide synthesis. This seminar will focus on the risk assessment of nitrosamine, specifically in solid phase peptide synthesis. And we will discuss the quality approach that we adopted to do this risk assessment, which is a failure mode and effect analysis. And we will discuss for a second a very brief section regarding nitrosamine quantification and the tools that are out there that we have selected to quantify nitrosamine contamination. And then we will go to the conclusion. So in 2018, news came out that there were some recall in the industry. Specifically in 2018, lots of the drug valsartan, which is an angiotensin receptor blocker, were recalled due to a contamination of a group 2A carcinogen, N-nitrosamine. And in the same year, other angiotensin receptor blockers, specifically arbesantan and losartan, were recalled for contamination of 2-nitroso and methylphoraminobutyric acid. These recall were for multiple suppliers of the drug product. And also the other thing that became apparent at the time was that multiple drug manufacturers, active ingredients, were involved with this problem. In 2019, lots of ranitidine were recalled because, again, NDMA contamination. This was a major recall that affected multiple brands. In 2020, lots of the drug nizatidine were recalled again for NDMA contamination. And in the same year, the drug rifampin and the drug rifampentine were also investigated for contamination of methyl nitrosopiperazine and 1-cyclopentyl for nitrosopiperazine. But this investigation determined that the level of these contaminants in the drug itself was not of concern, meaning that it was below the level that would cause a problem in human beings. Then in 2021, the drug metformin, which is used for diabetes control, only the drug that were used for the extended release of the drug product were recalled, also in this case for NDMA contamination. And in 2021, the drug varenicline, different lots were recalled for contamination of N-nitroso-varenicline, which was the first example of a drug recall where the nitrosamine was present in the structure of the drug itself. Then in 2022, the drug propranolol, which is a drug used for the control of high pressure, was recalled for contamination of N-nitrosopropranolol. Also in this case, the drug itself, the active ingredient itself was contaminated, was reacting with a nitroso compound to form nitrosopropranolol. Now, in this most recently, I think a few months ago, the combination drug quinapril hydrochlorothiazide was recalled for contamination of N-nitrosoquinapril. And just last week or 2 weeks ago, orphenadrine citrate was recalled for contamination of nitrosoorphenadrine. So as you see, this is an ongoing event and multiple drugs are affected. Now, let's try to understand what's the source of this problem. Secondary amine in presence of nitrates and high temperature and acidic condition can generate nitrosamine. This is the fundamental reaction that we're talking about. Now, also tertiary amine can generate nitrosamine by decomposition of the tertiary amine to a secondary amine. Now, most of the events that I've discussed so far involve N-nitroso-dimethylamine. And the question is, when this problem was investigated, the result on this investigation correlated the presence of the nitrosamine to the fact that dimethylformamide was using as a solvent in all these events. Now, I told you that a secondary amine will react to generate nitrosamine, and dimethylformamide is actually not a secondary amine. But what happened is, in high temperature, you have the composition of the solvent to generate dimethylamine that then become the substrate of this reaction. So we have to be cautious because dimethylformamide is one of the primary solvent used in solid phase peptide synthesis. Now, what happened at the time of this event was that all the agencies worldwide reacted to this recall. Specifically, the European Medicine Agency was the first to act, and in September 2019, generated a guideline for the authorization market holder that directed them to investigate the risk of nitrosamine contamination in their drug product. Specifically, the request indicated a very precise path. The first step in this investigation would be a risk evaluation of having nitrosamine through use of certain reagent, use of certain condition. After this investigation, this risk assessment was completed. In case the risk assessment had determined that there was a risk for nitrosamine contamination, the manufacturer or the market authorization holder were required to conduct analysis of the lot produced to determine if actually nitrosamine of the specific nitrosamine that was identified as a risk was detectable in their drug. And in case this nitrosamine were detected, the market authorization holder were supposed to inform the authority and initiate evaluation of the changes to reduce or eliminate the risk of nitrosamine contamination. And obviously, because these are drugs on the market, to proceed with a evaluation of the documentation and revalidation of the process to prove that the changes they were proposing were not affecting the quality of the drug. And in the initial request, the company were supposed to self-report with the result of step 1 and 2 and inform the authority if they were planning to initiate changes. Then in June 2020, the MEA also published a very interesting document, which is lesson learned from presence of N-nitrosamine impurities in certain medicine. I recommend that everybody who is looking into this issue for their product to read this document. It's very extensive, more than 100 pages, but at the end, it gave a very nice overview of the issue of nitrosamine. You will find out that this is not a new problem. It was already detected in the 70s, and back in the 70s, there was effort to reduce this risk. The industry probably forgot about this very important lesson from the 70s. And then in February 2021, the MEA published another document where they extended the guidance of nitrosamine contamination to biological products. The first guidance was only addressing problem for chemically synthesized API. The new guidance say that in the future, also biologic are in scope of this evaluation of nitrosamine contamination. But additionally, the MEA suggested a path how to control this nitrosamine contamination, suggested that they should be managed based on ICH M7 guideline in principle and considering a lifetime daily exposure to calculate acceptable limit. How the US reacted. So in 2020, in September 2020, the FDA published a document called Control of Nitrosamine Impurity in Human Drug, Guidance from the Industry, that was then revised in February 2021. Also in this case, the starting point is a risk assessment and confirmatory testing of presence of the nitrosamine of concern in the final API if this is or drug product. And again, if this risk is present, the manufacturer of the drug should consider changing to the process to reduce this risk. And these changes should be submitted as regular changes to drug master file with the existing path for this type of change to process. Then in November 2021, something that is interesting for people who is dealing with drug product more than people dealing with drug substance was that the FDA published a document called Addition of Risk Mitigation Strategy for Drug Product, which the agency suggested that there are certain additive and excipient that can be added to a formulation of a drug product that can reduce the risk of formation of nitrosamine during storage of the drug. What is interesting is in this document, the authority stated that the focus for this evaluation of nitrosamine risk assessment is on chemically synthesized active pharmaceutical ingredient and drug product. But there is that little sentence that say, but is not limited to this drug product, that opened the door to the fact that people dealing with biologics should also take this as a potential risk in their product. And finally, in December 2021, the United States Pharmacopoeia published a general chapter called 1469 on nitrosamine impurity, which provide, again, a general guidance about what are nitrosamine, where they come from, but also provide some suggestion regarding analytical method that can be implemented to detect nitrosamine. what about the rest of the world? So all of this recall started in one country and then evolved to different countries. And this highlighted the fact that we live in a world where drug products are produced starting with active pharmaceutical ingredients that are produced for multiple countries. So the regulatory agency of the major developed country in the world decided to initiate a collaboration that is ongoing in collaboration also with the WHO, so that events of nitrosamine problem in one country will be immediately communicated with the other country and will generate a network of information that is ongoing as we speak. So what are the nitrosamine of concern and where they come from? So the different reagents are precursor to the origin of nitrosamine, even if they're not secondary amine. And for example, I give you a list here of tertiary amine and solvent that contain amide function that can generate by the composition of by the combination of high temperature and acidic pH can generate secondary amine that are then in presence of nitrates, the substrate for the formation of nitrosamine compound. Specific solvents are very relevant to solid phase peptide synthesis because they are commonly used. The first one is dimethylformamide, that is the primary solvent used in solid phase peptide synthesis. And as already explained before, can decompose to the dimethylamine and in presence of nitrates can generate NDMA. The second solvent that is very commonly used is N-methylpyrrolidone, which in high temperature and acidic pH then decompose and hydrolyze to linear form of the pyrrolidone and then react with nitrates to generate NBMA. And disopropyl ethylamine, which is a very common base used in solid peptide synthesis, can also decompose to either ethyl isopropylamine that can then react with nitrates and generate the corresponding NAIPA, or can decompose to D-isopropylamine that can react with the nitrates to generate NDIPA. And these are some of the reagents that we need to keep an eye on and consider in which condition they are utilized. Now, let's move for a second into how we make peptide at Bachem. There are two major approaches to making peptide. They are both used in our company. One is liquid phase peptide synthesis called LPPS, and the second one is solid phase peptide synthesis. This approach are different in the way you generate the peptide. The reaction medium in the case of liquid phase peptide synthesis is a solution, while in the case of solid phase peptide synthesis is an insoluble polymer normally that we define the resin, where the peptide, the growing peptide chain is crafted. In the case of liquid phase peptide synthesis, the synthesis strategy is a convergence of stepwise, meaning you generate first a dipeptide, then a tetrapeptide, and you join the peptide pieces by pieces until you obtain the final sequence. In the case of solid phase peptide synthesis, the synthesis strategy is a stepwise approach, meaning you add one amino acid at the time until you have extended the chain. In liquid phase peptide synthesis, you isolate every intermediate, normally by precipitation, and the removal of the reagent from the reaction medium before precipitation is executed by liquid-liquid extraction. In the case of solid phase peptide synthesis, on the other hand, there is no intermediate isolation during the synthesis of the product, and the reaction medium are removed by filtration of the resin, which is a big advantage in terms of possible nitrosamine contamination, because you are washing constantly away all the reagent and the potential nitrosamine form during the SPPS. So the quality approach that was selected by all the sites at Bachem to investigate the risk of nitrosamine contamination was an FMEA. And we decided to conduct this FMEA for each individual product because there are different reagents used for each individual product. What is the risk and how we can analyze that risk? We decided to modify the standard FMEA approach to be customized to this specific risk of the nitrosamine by calculating, first of all, 3 parameters. The first parameter is the probability of failure mode. So we define a statement and we calculate what the risk of that specific failure in the process. And then we calculate the probability of having or forming nitrosamine that can result for that specific failure mode. And then we calculate the probability that the resulting nitrosamine that we have generated by the failure mode are carried into the final API. The severity of each individual failure mode can be described by the severity number, which is the product of the probability of failure mode times the probability of having nitrosamine derived from the failure mode. By the risk of each individual failure mode is defined by the risk number, which is the product of the severity number times the probability that that nitrosamine generated enter into the final API. One of the important things is that you have now a tool that allows you to give a value to the probability. And we adopted a scale from 0 to 5, where the number 0 for the probability indicates that the failure is not possible, because, for example, the substance is not used. A probability of 1 indicates that there is a very low probability and the failure might only occur theoretically. And value 3 indicates a medium probability, so the failure can occur. And a number 5 indicates that there is an high probability and the failure is likely given the process or the process input. Now, the other things that I think is very important when you do an FMEA is you should set a priori what you're going to do when you identify a certain risk. So the risk number define the action that we're going to take. Between 0 and 1, there is no need to take any action. When you have a value, a score of 3, the measurement should be considered. You are not required to do any action if you determine that the level of nitrosamine are below the acceptable level for that nitrosamine. While if you have a score between 5 and 27, you are still required to make measurement. And no action is required if a measurement is below the acceptable level, but you should consider improvement to the process to implement a control strategy to assure that that risk doesn't get out of control. While a value higher than 27 indicates a very high risk, and you should both do the measurement, obviously, but also you should consider action to reduce this risk, which are not control strategy, but most likely changes to the process. When you do an FMEA, it's very important to organize a very diverse team because different expertise are necessary. So in our case, when we did this risk assessment for nitrosamine, we have experts from the manufacturing that are experts in the specific process that is for the specific product that has been evaluated. We have members of the quality assurance unit, and we also have members of the procurement unit, because as I showed you before, a lot of the nitrosamine risk is related to the presence of nitrates. And they are the one that can help us evaluating the risk through analyzing our vendor and the quality of our reagent and so on. So the preliminary activity when you do this type of assessment is you generate a bill of material for the product that includes all the reagent, solvent, material of contact that are used to produce a certain product. For us, this is very simple because we have an enterprise company solution that maintains the bill of material of all the product we produce. Then you move into establish the origin of the reagent and solvent. Where do we procure them? What type of contract are in place and what type of quality we have for this solvent? You then go back to your manufacturer and ask them to evaluate the risk for the material of containing either nitrosamine or nitrates. And finally, for the product that are produced internally, like our building block, we review the raw material bill of material and we discuss with the specific people who produce this reagent what is their evaluation of the risk of having either nitrosamine or nitrates into this reagent. Normally, the FMEA question you have to ask, which are partially derived from the suggestion from the European medical agencies, are there amine structure in the reagent and in the starting material? For example, the amine structure could be hidden into an amide function into the solvent, which can contain secondary amine. The second question is, are there a risk for nitrates to be present in each of the reagent that you use or solvent that you use? For example, may the reagent contain nitrates, and we identify one possibility, and I will discuss later, which is oxima. And one of the things that were stressed by the authorities to carefully evaluate the water you use in your processes for possible nitrates. And then finally, you have to ask the question, do the reagent or solvent, can they contain directly nitrosamine? For example, through degradation or as impurity. Now, this is an example of a risk assessment for one failure mode. I'm giving you the example of the failure mode. Solvent that are used in the process contain an amide group, which can contain secondary amine, for example, DMF and NMP. Now, the probability of failure is very high because we know we use this type of solvent in production of peptide by SPPS. But if you look at the process through evaluating the other individual risk, we arrive to establish that there is no risk of nitrosamine derivative, nitrosamine compound being present in the final API because of multiple reasons. One of them is that DMF constantly wash the product by SPPS and remove away any possible contamination. And there are other factors like the fact we use HPLC purification. Let's take a look at an example of a medium risk that was identified during our initial FMEA in 2020. The failure mode in this case was that the reagent may contain nitrates, for example, nitrates in oxima. The probability of failure is high because nitrates are used in the production of oxima. Specifically, ethyl cyanoacetate is reacted with nitrous acid to generate oxima. And the probability of nitrosamine resulting for the failure mode is low because at the same step where we use oxima, we don't use secondary or tertiary amine. But on the other hand, the severity number is still 5 and is high. Now, the probability that the resulting nitrosamine are carried into the API is low because the residual of amino or amido solvent and reagent and potential nitrosamine will be removed by multiple washing steps during solid phase peptide synthesis. And in addition, the product is purified by a reverse phase HPLC, which indicates that the small reagent or nitrosamine will most likely be separated because of different retention time with large peptides. So the risk number is going to be calculated as 5. So the risk of nitrosamine in the final API is does not require measurement of nitrosamine in the final API. But on the other hand, there is no identified nitrosamine that can be in the API. But in conclusion, there is a medium risk. And so what we decided to do was to perform testing of nitrates content in Oxima. Now, all the lot of Oxima in the company were analyzed for residual potassium nitrates and sodium nitrates and did not identify any level of this contaminant below the limit of detection that was that existed for that analytical method. But the beauty of a FMEA is that you can now calculate what's the recalculate the value after you have made certain decision. So for example, in the case of the follow-up FMEA for the process, what we decided to do was to establish a control strategy, which is now every lot of Oxima purchased and by Bachem is analyzed for nitrates. We specify predefined specification for nitrates. And this is now our control strategy for this specific risk. So now if you redo the FMEA and you look at that specific risk, meaning reagent contain nitrates, nitrate in Oxima, we can have a probability which can be defined as low because we have implemented a control strategy of Oxima. So in conclusion, this risk has been reduced by putting in place a control strategy. So get ready for the unexpected, nitrosamine quantification. As I told you, in case you detect a risk for nitrosamine contamination, one of the expectations from the authorities is that you quantify that nitrosamine in your final API. So while we were executing the evaluation of the risk, we were considering the possibility that there was a risk for certain nitrosamine to appear in our product. This was not the case at the end, but we wanted to have the method. So our QC initiated very early in 2019 development of a test method appropriate for the quantification of nitrosamine of concern in the final API. Now, they adopted a headspace procedure based on procedure C of GCMSMS platform, which is described in the European Pharmacopoeia. We generally validated for 7 nitrosamine, and we were able to obtain level of quantification between 10 and 100 ppb, and level of detection between 2 and 28 parts per billion. Now, what we also did as a test, we took some of our API and we spiked in the nitrosamine of concern to try to verify if the presence of the API will modify the limit of detection and limit of quantification. There was a little effect, and it was decided that if we will conduct tests of nitrosamine in the final API, we will conduct a validation and verification including the LOD, LOQ, using as a matrix the API itself. So that is on the table in case we need to do nitrosamine quantification in peptides. So in conclusion, with FMEA, we are able to control and avoid nitrosamine contamination in our API. There are a few factors that are relevant when you look at the problem of nitrosamine in the synthesis of API by solid phase peptide synthesis. First of all, the process with FMOC terbutyl strategy avoid acidic condition, because this is incompatible, not compatible with the raising strategy we are using. In solid phase peptide synthesis, you have extensive washes that constantly remove solvent from the solid support that most likely can exclude contamination of nitrosamine. And for all the product that will be produced by soy phase peptide synthesis, we conduct preparative FPLC purification, which further reduce the risk of carryover of nitrosamine into the final API. We only use chemically produced reagents and solvent, and we have a very reliable supply chain, which guarantee the high quality of our starting material. We have identified a risk, which is the possible source of nitrate in Oxima, but we also have put in place a control strategy to limit this risk to almost non-existent. And I want to point out that very often we mention the fact that secondary amine are a source of potential reactivity with nitrates to form nitrosamine. On the other hand, in soy phase peptide synthesis, secondary amine are very deleterious to the chemistry itself. So we have always had a very tight control on our tertiary amine for the absence of secondary amine, because that will induce an early removal of the FMOC protecting group and can cause insertion and other problem in the chemistry we are using. So we are helped by the chemistry itself to keep a tight control of certain of this impurity. So what I can tell you is that Bachem is a company that operates worldwide, and this nitrosamine is a worldwide issue. So we are able as a global company to react quickly to any new potential risk for our API. An in-depth analysis of all the components that goes into the preparation and the process that are used to prepare active pharmaceutical ingredient is the key to guarantee that our product meet the quality that is required, specifically in this case, the absence of nitrosamine. We identify FMEA as the ideal tool to conduct this complex and multi-parametric risk assessment. I think you will appreciate that both peptides that we produce are made by SPPS, and most SPPS processes share the same materials a unit operation. And this provides a synergy for the assessment of risk of nitrosamine contamination. And we have finally executed a very efficient and flexible risk assessment, and we are able to apply that risk assessment to the product that required this assessment. Back to Leo.
All right, thank you, Matteo. So we have entered the live portion of our webinar, and we'll start with a poll question. Have you previously conducted nitrosamine risk assessments on your projects? You should see radial buttons in front of you, and please select the best answer for you. All right, then, it looks like, a majority of you have performed the nitrosamine risk assessment or are planning to. Great. And with that, let us now move to the question and answer section. Okay, great. So our first question is in regards to coupling reagents. So Matteo, is there any other coupling reagent of concern that carries nitrite-related impurities?
So we had, in the limit of the cases that we had to analyze, there were other coupling reagent, or let's call it coupling additive. I think the question referred to item like HOBT, or which is an additive to coupling, or for example, I don't know, TBTU could be one example. We did not identify in the way the material were produced a risk for nitrosamine contamination. But obviously, I think there are maybe approximately 300 different coupling reagents. There is a very nice review on coupling reagent by Professor Albericio, and we did not analyze all of them. We only look in the ones that were relevant to our processes.
Okay, great. Matteo, we have another question here with regards to the process chemistry itself. Can you comment on surfaces that come into contact with the API and whether there has been some study or are there studies done that evaluated the impact of the manufacturing equipment or the primary containers?
So the part of our FMEA did cover the manufacturing equipment contact surface. And these were, in case of making peptide, very often you have either stainless steel, which is most of the plumbing through the purification process, for example, as an example, but you also have tubing that are used to transfer liquid and material. And we did analyze all of them, how they were done, what were the plasticizer included, and we did not identify a risk for this type of liquidity. We also look into the filtration device. We produce material that are sterile. Sorry, we produce material that have a control bioburden. So we use very often filtration devices. These are filtration devices that are standard in the industry. It's nothing special. But we also did we evaluated carefully the compound or the material that constitute this filter. You have to realize though that when you talk about API, these are small surface compared to the quantity of the API produced in bulk, as opposed of something like this filter versus preparation of the final drug product. And also there nothing. And finally, for the material that we use in my site for storage of the API, we have glass bottle mainly, different sizes and with a specific closure system. And also in this case, the glass was not considered a risk, the type of glass we use, and the container closure was not considered a risk. So in that sense, for our process, we did not identify any risk, but everyone who does a risk assessment has to look into their specific product, so material of contact, and make that assessment.
Okay. Another question we have is more on the overall CMC strategy side. So Matteo, when should the risk assessment be completed? Before PPQ or after PPQ?
The authority has not defined this risk assessment during clinical trial, not to my knowledge at least. So we don't see the authority in this moment asking about nitrosamine contamination for material produced for clinical use. I think they are going to ask about this for material during the review of the drug master file of the application from the sponsor when they seek approval, meaning that you have to be in the position to provide this risk evaluation when you file. You can try to do it after validation if you, like in our case, you have a very tight control of the process and you have a process that doesn't introduce unknown entities regarding what we have been doing in terms of reagent and processes. And there are no special. And I think it would be safe to go into PPQ without having done the risk assessment. But as soon as your product contain a reagent, for example, that are not classical reagent for peptide production, things like PEG, for example, you want to know if the people who produce PEG, if you're going for a PEG conjugated product, have evaluated their PEG production for risk of nitrosamine. Now, if you don't start this request early and you use a PEG from a supplier and you make an API, at the end, if the moment you go and ask this confirmation from the supplier, and the supplier tell you, I cannot guarantee you nitrosamine, the lack of nitrosamine or nitrates in my product, then you will have a difficult time, because you will have to put in place the control after you have completed your PPQ, which could be a risk that you need to change something, and that change may push you to do a new validation of your process. So depending on the complexity of your product, it might be earlier or later of the PPQ.
Okay, cool. Another question here, Matteo, on the starting material side. As we know, many peptide drug candidates are now incorporating unnatural amino acids, including custom synthesized residues. So from a risk mitigation standpoint, Matteo, especially for these de novo amino acids with very unnatural side chains, would it be best for some of these unprecedented building blocks to begin their nitrosamine assessment?
Yes, I would say that that you will try to establish with your supplier of the material if they have what's the route that they choose to produce this product, and if they use anything that could introduce nitrosamine or nitrates in the process, and you want to choose a supplier of this raw material that is able to answer some important question. One of the important questions, in my opinion, is, do you use recycled solvent? Because some of the event of this nitrosamine contamination seems to be related to the fact that in the two countries where this product seems to be originating, I'm not sure 100% is only there or is there. The two countries are in China and in India. And there was a practice to recycle DMF, for example, derived from different processes. So you will send your DMF, you will receive back the DMF that was recycled, and that DMF was mixed with DMF from other processes. And so you think, I have no problem because I don't use any nitrates, my DMF is clear. You send it over, and then somebody else has used nitrating agent and other things in their DMF. And the amounts that constitute a contamination are very, very small. So you will probably, especially if you don't look for them because you're not aware that somebody else has used them, you will not see them. That's why I insisted during the presentation that we only use solvent that are freshly synthesized and not recycled through distillation, filtration, or other procedure like that.
I see. So Matteo, I guess some efforts to improve or increase the green character of processes may in themselves create additional risks and implications for nitrosamines, particularly those that could decompose into secondary amines, right?
Exactly. So you have, if you were to think, I want to operate with DMF and recycle my DMF, you have to know exactly what that recycle looks like. If you require, you could think about, because I don't use nitrates, so you can think about recycling, having an agreement with a company that guarantee that they will recycle only your DMF, or they will keep your DMF recycling separate from somebody else recycling. That could be a way to be greener using recycled solvent. It's all a question of put the weight in the quality of that recycling. Or you could decide to recycle in-house, and when you recycle in-house, you have a plan to recycle, and if you don't use any potential agent that can contribute to the formation of nitrosamine, then your recycle become very safe. It's a question of controlling the process and know what you're looking at. So you need people who has a certain understanding of the issue associated with this problem.
Okay. And then the limits, the exposure limits have already been defined by toxicology studies, right? more or
So
less?
there is for the major class of nitrosamine of concern, there was the initial series of components. The authority has given limit. You can find this limit in different documents. Now, I just saw a presentation last week that was extremely interesting, where some people were starting to look into why a nitrosamine is toxic. And the interesting thing is not a direct transfer of the nitric oxide from the API into gene or into DNA and alkylation of the DNA. There is a decomposition of the nitrosamine with an alkylation that with a transposition that involve an attack to the alpha proton to the nitrosamine itself. And for example, you have situation where that decomposition cannot occur or is unfavorable. And so the discussion was rotating around the fact that not all nitrosamine are by default genotoxic or mutagenic, and that there should be a movement or an attempt to put some scientific rationale behind this nitrosamine. And the point they were making is, so different nitrosamine may have very, very broad range of mutagenicity. And so in this moment, because of this lack of a systematic approach, they were discussing the fact that maybe MS tests could be used like they're used for other mutagenic compound to evaluate the risk of certain and the dose of certain nitrosamine. And this maybe can be done without doing the full study, but using rational evaluation of the structure of this molecule. They were making the point that maybe some of the value that are out there are extremely conservative, and maybe an evaluation of these numbers with a slightly different toxicological mentality could be useful to alleviate some of the fear that we have now. We have a lot of fear about nitrosamine, and people is taking a very conservative approach, or it should be, but I think you overcome fear by understanding the risk and doing the proper work. I'm not a toxicologist, but I believe there are toxicology out there that could maybe, there are group study, the USP has a group study on, an exchange of information between different people who work on this nitrosamine issue. And these people can help to relieve a little bit the fear that all the major pharmaceutical companies have in this moment for this risk.
Okay, Matteo, some questions now more on the late phase or post-marketing approval side. Has Bachem completed nitrosamine risk assessments for its generic APIs?
Yes, the EMEA directive was very clear, and because a lot of our generics are involved in the are on the market, we did complete risk assessment for, I would say, most of our generics, or at least there are different sides of Bachem, but I think the other side did the same activity for other generic because we were listed in the marketing authorization. And the European Medicine Agency had a very tight deadline. So this whole process started because we needed to act. And the generic were the first in the line. Most of the people who buy our generic came to us and say, we need your help to answer the question for the authority. So generic have been evaluated.
Okay, and another question more on the post-marketing side. If we wanted to file an ANDA or an ANDA using a Bachem generic product, how can Bachem support our ANDA filing for nitrosamine?
I think our quality people and the business development people can support anybody who has this need. If you are filing, I think we can, with our material, we will be able to provide you the information that you need to file if the question is raised by the authority when you file. For the API, you have to remember that I offer you a point of view today on the active pharmaceutical ingredient. The authority don't stop at the active pharmaceutical ingredient, because there have been cases where the print ink in the blister of a drug was involved with the formation of nitrosamine theoretically. And there is, as I say, there's this directive or this document from the FDA that goes into the detail of giving the suggestion for the formulation of the API, which means that they believe that the formation of the nitrosamine, it's also in the associated, for example, with the excipient use. So we can support you for the API, but I think you will have to do your evaluation with your formulation company regarding your excipient, your process, the filing, and everything else. So we can help definitely our customer for the API that we produce. We have been doing that actively.
Okay, great. And with that, it looks like we have gone through all of our questions. I'd like to thank the audience for attending, and thank you, Matteo, for this very informative and helpful, hopefully, to our audience webinar. You can find out more information about the content today, as well as other technical topics, by visiting our various content channels, including on YouTube, as well as our website, which has many white papers, as well as other technical information that can hopefully help you choose and design the best strategy to develop your new chemical entity or your other new drug substance. There are other ways to contact us. Please feel free to send us an email or write to us. And with that, I'd like to thank everyone again for attending our audience, and have a nice day.
Have a nice day, everyone.
Bye.